So next off, I'd like to introduce our next speaker, Dr. Tan Shen Jiang. He's a Senior Research Associate of the University of Cambridge's Department of Hematology, where he's done extensive research in linking defective ribosomes assembly to myelodysplastic syndromes and functions of factors involved in cell proliferation and disease development. He is also the Research Associate at the University of Oxford's Department of Zoology and has published in numerous journals. Please give it up for Dr. Tan.
Hi, good morning, everyone. First of all, I would like to thank the host, Mr. Chen, and also all lab group, and to invite me to here to present some of my work in the lab, and also we want to hopefully through this event to create more collaborations between Cambridge, China, and Malaysia. So I'm Shen Tan. I'm a senior research scientist in Cambridge. So I'm currently affiliated to three institutions, like a Department of Hematology, and also the Cambridge Institute for Medical Research, and also the Cambridge Stem Cell Institute. So the basic research we are doing in the lab is about ribosome biogenesis and cancer. So what's ribosome? I think everybody knows that in our biology book, the ribosome is named as a protein factory. So it's a big molecular complex pre-assembled in the nucleolus in the cell, and then after a series of assembly events, it will, submit to pre-40s and 60s and transform from nucleus to the cytoplasm. And after a further step of maturation, the 40s and 60s can join together and then start to read the genetic code, or we call it decoding, and then start to synthesize new proteins through this peptide channel, and then it will bind into the ER, and then all those new synthesized proteins, after a series of quality control, it can be transferred to the cells to perform their functions. And due to this importance of this cell machinery, so three people shared the Nobel Prize at 2009. And why it's so important in the cell? Because we know in our human cells, each cell contains about 10 million ribosomes. And every minute, so this is reconstruct structure of the HeLa cell. So every minute through this nuclear pore, about 15 to 20,000 of ribosomes submitted has to be transferred from the nucleus site to the cytoplasm and to do the further steps of maturation. And it also has been found that recently in 40% of human cancers, this patient carry about genes mutated in ribosomal proteins. So that's why in our lab, we're trying to find out what's the mechanism of this ribosome assembly and how those mutations happen in ribosome assembly factors or ribosome proteins that will lead to certain diseases. And these diseases, most of them are MDS and AML, and now we give them a new name. It's called human ribosomal palsy. And there has been a few types of ribosomal palsy has been published or recognized by people. So there are some acute ones and also inherited ones. So like DBA, Short-Diamond syndrome, Tracheal syndrome, X-linked dysclerosis, and Congenia. So all those ribosome palsies, they carry, the patients carry mutations either in ribosome proteins or factors associated with ribosome assembly. And finally, all these patients will develop to either AML or MDS. And the lab in our lab is most focused on this disease we call Short-Diamond syndrome. So it is an autosomal recessive disorder caused by the mutations of the gene called SBDS. And most typical syndrome or typical clinical phenotype of the patients they've got is they started with pancreatic insufficiency, and also later on they'll have aplastic anemia, MDS, and at certain stage, so mostly like in the early 30s, about 60% of the patients will develop to AML. And most of the patients, when they are born, they got this very severe growth defect, and also like the skeletal defect as well. So in our lab, we have found this disease caused by the mutations in this gene SBDS, and we have already found the function of this gene or this protein is involved in the last step of the big subunit of the ribosome 60s. So at the last step of the 60s maturation, there is one factor called EIF6. It gets stocked on the surface where the 40s were going to join the 60s to form the functional 80s. So this factor has to be released. And the function of this gene, SPDs, or the protein, is to collaborate with another GTPase EF1 to release EF6 and then to facilitate the binding of 40s and 60s to form the functional 80s, and also will prevent the rejoining of the 40s of the EF6 back to this 60s after the dissociation of 80s. So in the lab, we use lots of animal models and also the most recent advanced technology we call cryo-EM technology. So by purified proteins or ribosomes from the cell, and we can detect the structures and then reconstruct the whole process of the ribosomal assembly. And so far, we found that how this gene is sitting on the 60s is so they're sitting on the middle of the 60s and then have some amino acids point to the peptide external. So this is the terminal where the new censored peptide has been passed through and back come to the ER. And if this process has been affected, so you can imagine that the translation cannot be done properly. And this movie will show how these two proteins, which is SPDS and EF1, happen together to kind of release or we call it kick off this EF6. And after that, these 60s can join the 40s to form the functional 80s. And if there's something wrong with this gene, we know that patient will cause this disease called SDS, and we also did some biochemistry and experiment showing that. So this is to isolate all the ribosomes in the cell. We call it Poisson profile. We see that so if we isolate from the normal cell, we've got 40s, 60s, and 80s, and some poisons. While if in the patient, we can see the active 80s where the protein has been synthesized is much lower than the control. And also if we monitor or checking the in vivo translation, we can see that the patient, they got much lower relative of those protein synthesis. And so we now also found that this disease, as a short-term diamond syndrome, is not only caused by the mutation of SPDS, but can also caused by the mutation of EF1, which is another partner that collaborates SPDS to release the docking factor EF6. And patients carry EF1 mutations have the similar phenotype in the cells, where you can see much fewer ATS and also increased of 60 subunits of ribosomes, and you can see the translation is done as well. So hopefully, through those animal models and also human patient samples, we're trying to figure out how the maturation of 60s has been quality controlled in the cells, and then by defining those mechanisms of this 60s maturation, we're trying to finally find out how the defect in the release of EF6 and also maybe the global translation defect, how that will cause P53 activation, which we have been found in most of the patients of ribosome palsy, and also those DNA damages caused by replication stress, which cause the genome instability, and finally it causes MDS or leukemia aggregation. And so these are the basic research happening in the lab. But what we're trying to do is, we hopefully, through what we found in the lab, we're going to create the opportunities for some novel therapeutic strategies for the treatment of the disease. So we're definitely including the traditional target drugs or medicine, and also at the same time we're going to introduce the new cell therapy strategies through the treatment of those diseases. So currently happening in the lab is, because we found that the release of EF6 is very crucial for the last step of 6-stage maturation, and also through a series of genetic screen, we found lots of EF6 suppressors. So basically, we found 24 point mutations of this protein, which most of them sit on the surface of the protein EF6. So it's that surface. So they will kind of abolish the binding affinity of this protein to the ribosomes, but at the same time, they can bypass the defect of cells which lack of SPDS or EF1. And currently, we are doing lots of studies in cells. Well, we have done lots of structural biology studies already. We found that those EF6 suppressors, they can bind in to the 60s, and also they can help the release of EF6 in those patients' cell extract. And now we are testing those. We have found lots of small molecules which mimic the function of these EF6 suppressors, and we are doing some cell studies. So basically, through those small molecules in the cells, and monitor if they can increase the global translation a lot. So this is showing what, this is one of assay we're doing in vivo measuring or monitoring the translation. So we can monitor the translation by using a pyrimidine analog. It's called opopuro. So stay in the cells and then monitor how the signals has been changed. Basically, those signals will indicate the new synthesized peptide, and then we can do in using a high throughput microscope assay and also statics, we found that some small molecules or maybe potential drugs at certain dosage, they can increase the translation of the patients. So this is using one of the patient cells. We do the assay and they can, so some of them, they can increase the global translation, but some of them, they cannot. So this is the target drugs or target medicine, but also we would like to try this therapies for leukemia. But we have already know that the treatment of leukemia, which probably be the first successful use of cell therapy, which is stem cell transplant, right? And we know that if patient got leukemia, they can transplant donors bone marrow, which mostly is transplant the hematopoietic stem cells. But this method got lots of limitations, like the limited or restricted source of bone marrow donors, and also they got GVHD reactions, and also they got age restrictions. So for most people, if they are diagnosed as leukemia at age of 40s or 50s, it's very difficult for them to do the blood transplantation. So opportunity or potential way to bypass this problem is that we can use the iPS-based technology. So as Professor Gu has mentioned a lot in his talk about this iPS technologies, which is also so like John Gurdon. So he's retired already, but he's still the PI of the Cambridge Stem Cell Institute. So they won the Nobel Prize in 2012, and also currently in the institute, there's still lots of research still going on based on this technology. And because we are in the Department of Hematology, so we want to know how we can bypass those problems of the traditional blood stem cell transplantation. So we have some collaborations with Professor Gu's lab and also George Daly's lab in Harvard. So trying to find out a way, so using these iPS cells to generate or we kind of direct differentiation of iPS cells into induced hematopoietic stem cells. And then after a series of quality control and safety experiments, then we can transplant to the patient. So currently this work has been done in the animal models and has been tested. It's safe and is effective. And the next step is we're trying to, we're currently still in the process of application of the clinical trial in Cambridge. So it's by the collaboration with Olaf and also George Daly through America. So that's the first part is the stem cell based cell therapy. So another part of cell therapy will be like Professor Gu just gave you guys a very informative presentation about the immunotherapy. So this slide showing how the CAR T cell works, but we also from lots of studies and also Professor Gu just mentioned the lots of restrictions about CAR T. So I don't want to mention more. I just want to go through the next slide, which is those technologies based on NK cells. As Professor Gu mentioned already, and also there's lots of studies and still currently lots of clinical trials still going on, is that so before the CAR NK technology, people are still using the normal NK cells to treat leukemia. So the mechanism is very simple. So you isolate or purified NK cells from patients, and then do the activation through a few cells and interleukin 2 or 5 or 21. So as some doctors in the audience have mentioned that in some studies it mentioned about after the transplanting of NK cells, some patients do have same like the GVHD or CRS syndromes. And later on, through the studies, they see that the reason of that is just because their NK cells is not pure enough. They contained a few T cells. That's why it caused the GVHD reaction and also the CRS. So our next step is because, so if we can find a specific tumor cell antigen, it will be much helpful for us to design or engineer the NK cells to target the tumor cells. But for some tumors, if we haven't found a specific one, or if we found one we haven't validated that is applicable or effective in animal models, we cannot use that. So I will give an example. It's here. So like this leukemia cells, they have CD123 and CD33, both markers. Okay, maybe they're both specific to this, but later on, through studies, people found that this CD123, they can also present in some lymphoma cells and also in a small group of hematopoietic stem cell progenitors. So if we target CD123, that will definitely cause GVHD or CRS syndromes. So the only thing is that so far we have found that the most safe way is target CD33, so not 123. So for the future plans and collaborations, so as I said, it's also it's very important still continue to do the basic research. I mean, to find out the mechanisms of different disease, and after that, if we found a specific marker or specific antigen, so we have more specific targets to treat the disease. So in our case, it would be we need to do more research trying to elucidate the molecular mechanism of ribosome assembly, because during the ribosome assembly, there are more than 200 factors involved in the process. So it's still a long way to go, but so far we have found some important findings. We can use that to target some group of diseases. And also, we need to validate specific cancer antigens and markers for CAR-NK. And then after that, we can start the clinical trial of cell therapies. So in summary, it's just two simple messages. Is that first, we need to do basic research to reveal the molecular mechanism of disease, and also we need to combine target medicine and cell therapies for the control of diseases. And at last, I would like to thank the support from all the institutions and also the funding bodies for our research, and thank you for your attention.
Thank you, Dr. Tan. I'll be opening up the floor again for another session of Q&A. Please raise your hands if you have any questions. All right, if there's no other questions, very well. Oh, sorry.
Actually, the CD123 and the CD33, you were saying that the CD123, if you tag them to the CAR-NK, you'll be less effective and there's more resistance. Is that true?
It will I won't say it will be less effective. It will be more effective, but you may have severe side effect.
If you target to the 1, 2, 3.
That's right.
What was the side effect? Is there a cytokine storm if you do this 1, 3, 3 targeted therapy?
Well, we haven't tried that in humans. It's just we found that through the studies in mouse models, and in mouse models, some of them they don't like. I think it's in the mouse models. Some they have CD123 in a group of progenitors of the hematopoietic stem cells. But we know that in human cells, there's some lymphomas, they are CD123 positive. So if we target CD123, we may target this lymphomas. So that may cause problems. So because of this concern, so we think it's safe not to target CD123 at first.
Yes. So when you do, so all these studies is a murine study out of the mice on the mouse model that you publish. Is this all this is mouse model? Yes.
Yes. Well, that one is not our research.
Okay.
Not our research. Just I mean we check the research and how people design their studies, and we found so okay based on people's study, we see that might be a problem. So we need to, like I said, if we want to develop or engineer the NK cells, we need to validate if that's true or not, right? As people mentioned, that may be a problem, so we need to confirm that in our research lab of the next OLAF lab and also in our laboratory. Okay, after we validate, it's there. Maybe they're wrong, right? But we have to validate that, and then we can do the next step.
Yeah, for the multiple CAR T treatment in the world done already, then later they found out there's resistance because of the PD-L1, CTLA-4. Would NK cell treatment later, then you will have also resistance. Have you seen resistance later or due to the checkpoint? Is there any checkpoint resistance possible in NK cell therapy?
I haven't found any. Yeah. So
in these centers here, is this already done on human? I mean, CAR NK against CD33 on leukemia patients.
This is one of our project still going on in the lab.
Okay, thank you.
Yeah, as Professor Gu mentioned, we started from the prostate cancer first, but because we are in the Department of Hematology and we want to target leukemia as well, so we're trying to find out which is the best candidate or target. So that is the one we found, and then we are developing the protocols to do that.
CD19 and the CD22, and the CD38 we are also testing, and this is one it's an ongoing project. We think whether target 33 or target CD213, and it seems like the one 213 not because it's presented in the kind of the stem cell or the stem cell derived cells. So in that point, we not target. So at the moment we have is CD19, CD22, 22. Yeah, this one.